Hello, and thank you for joining us today for this webinar, a comprehensive guide to optimizing your small molecule biology development. My name is Andrew Warmington. I'm the manufacturing editor at Stoutline, and I'll be your moderator. The webinar is brought to you by Scrip and sponsored by Illyri Bioanalysis. Before we jump into the presentations, let's go over some housekeeping items. First, if at any time you're having technical difficulties, please click on the Test Your Connection button, and this will take you to a connection checker and FAQs page. There's also a Chat Now function to contact the support team for assistance. If you want to enlarge the presentation, just click the Maximize icon or drag the lower right-hand corner of the presentation window. There'll be a Q&A at the end of the presentation, so if you have questions, we want to hear from you. To ask our presenters a question, simply type it into the question window on the bottom of your screen and then hit Submit. We'll be answering as many questions as possible during the Q&A session, and if there are any extra, we'll try to answer you after the webinar via email. And because we know you want to listen to this webinar again, it's being recorded and will be available for you on demand in about 48 hours' time. I'd now like to introduce our speakers for today's session, Troy Volker and Corinne Ramos. Troy Volker is the Director of Laboratory Operations at Illyri Bioanalysis in Salt Lake City, where he leads all of the lab divisions, including method development, method validation, instrument operations, and production groups. Troy obtained his PhD in synthetic organic chemistry with a postdoctorate in medicinal drug development and drug metabolism. He's been working in the bioanalytical industry for over 17 years, and he has developed assays for large and small molecules with extensive experience on both triple quadrupoles and high-resolution accurate mass platforms. He's the Illyri subject matter expert for oligonucleotide bioanalysis, in which he's led development and validation for the past 10 years. Corinne Ramos joined Illyri Bioanalysis in 2020 as the Director of R&D to develop clinical programs to support innovative research. As a professional with more than 20 years of experience leading innovative research and clinical programs, Corinne is known for driving clinical developments to successful commercialization. She holds an MBA from the Johns Hopkins University Carey Business School in Washington, D.C., specializing in finance, and a PhD from the University Paul Sabatier in Toulouse, France, specializing in molecular biology. And with that, I'd like to hand things over. Troy, the floor is yours.
Thank you, Andrew. So thank you all for joining us today as we talk about bioanalysis and optimizing your small molecule drug development strategy. So the first thing that we would like to dive into would be a step-by-step approach to a rugged method. This starts with method development. In this phase, we'll talk about mass spec tuning, HPLC separation, sample preparation, stability, matrix effects, method development to method validation, and method validation into sample analysis. So everything starts with mass spec tuning. If you don't have proper conditions, you're not going to have the sensitivity you need, and you may not have the robustness for the assay to make it through a sample analysis method. So sensitivity optimization should be done for at least 2 MRM transitions per analyte and internal standard. One can always be used for a qualitative measure and the other one being used for a quantitative measure. We like to follow as many as possible in case there are issues that arise with our lead transition, and then we still have collected information for other transitions. You should tune in both acidic and basic pH solutions. You never know really which is going to provide you the best sensitivity or selectivity, so I would never go in assuming either acidic or basic would be the best option. I always like to use at least a few organic solvent modifiers, whether that would be ammonium bicarbonate, ammonium formate. There are hosts to try. Once again, you just want to check off the boxes as you start this progress into method development. A second part is you never want to assume that something's going to be positive or negative. So checking both at this point in time is your due diligence in order to ensure that you do check those boxes also. And I know everything pretty much works in ESI mode, and APCI is kind of a forgotten source, but you should check APCI. Since very few things work in APCI, when yours does work in APCI mode, you often eliminate a host of problems if you can start your method development in that source mode. For HPLC separation, retention time characteristics should be studied as a function of both acidic and basic mobile phases. Often people think because they are in a positive ion source mode that they need to use an acidic mobile phase, but honestly, basic mobile phases can still provide great sensitivity and often provide better separation for other basic compounds. You should look at your retention times as a function of the organic modifier. So you really want to aim for a K prime of 2 or greater. We often do assays that have multiple analytes within that assay. So you have to focus on all of them being retained on column and then selectively eluted off of the column, looking at both peak shape and overall sensitivity. The determination of your chromatographic mode behavior, we like to do breakthrough experiments in our lab, where we will look at how each function of the organic in terms of percentage will elute off the analytes of interest from the column. This is done in order to ensure that we do achieve that optimal K prime. And then you'll also want to ensure that there are the separations of the compounds of interest from both endogenous and from themselves. We all know about phospholipids and the impact that they can have on an analysis. We often don't know what else may be in the matrix. So just looking at overall separation is important. So for sample preparation, I mean, there are 3 main modes of looking at this, and we'll go into the pros and cons of each one, both protein precipitation, supported liquid exchange, liquid-liquid extraction, solid phase extraction. But the main thing I want to discuss on this page is to ensure that your aliquot size that you're going to use for the assay will work with the sample collection procedures. You don't want to go into assay method development with 100 microliter aliquot when you're only going to be able to get 25 microliters from the actual sample. So going back to the protocol, talking with your sponsors to ensure that what you're developing will work with the samples is absolutely a must. So for protein precipitation, you know, the pros is it's very high throughput. It works with pretty much anything when we're talking about small molecules. It's cost efficient. You know, that's very low error propagation in the fact that there aren't many steps that are done, and it's very amenable to a very large set of compound classes. On the back end, the cons are you do create a very dirty sample. So going back to that previous statement of making sure that you are separated from other type of endogenous matrix things, if you go through a protein precipitation, you've made that step extremely important because those entities are going to be present within your sample extract. It's non-selective, so you're not going after just your compound in this type of extraction. You're going after all of them that are going to be solvent or soluble within your crashing solvent. It does not perform well with increasing molecular weight. So the larger they get or the more protein bound those compounds are, typically this type of extraction would not be your choice. For supported liquid exchange, once again, it's very high throughput. It also creates very clean samples on the back end as you are really just pulling out things that will be soluble in the organic phase that you're using to separate them into. And it is also very low error propagation in the fact that there's also still very few set. It's more selective than a protein precipitation extraction in the fact that you're not taking everything that is soluble, but rather things that would be of the right pH and will phase transfer into your organic solvent. The cons is the samples can, your sample recovery can vary from well to well. This is something that can be mitigated by going very slow with regards to your elution and letting gravity do most of the work, but we still can see some variance. There are limited bed sizes when you're working with high throughput. So 200 or 400 microliters are the 2 main options that you can work with. So once you get into those very high aliquots for very sensitive assays, typically you would have to move towards liquid-liquid exchange. This also does not perform well with increasing molecular weight. So a liquid-liquid extraction really has pretty much the same pros with regards to selectivity. You also get more consistent recovery from sample to sample in the fact that you're shaking these vigorously, so that phase-phase transfer is much more clean with regards when comparing to supported liquid exchange. And again, you're creating very clean samples. The con, though, it is not high throughput. These are all manual-based extractions. They take a long time to process. But on the other hand, you can use a large sample aliquot volume. So although it's listed as a con here, that's actually a pro. This also does not perform well with increasing molecular weight. So a solid phase extraction, very clean samples, very high throughput. You get a large choice of extraction phases. You have weak cation, cation exchange, weak anion, anion exchange, hydrophobic, hydrophilic types of separations. So there's a very large toolbox that you can work with here. It works with both large and small aliquot volumes, and it can be extremely selective. The few cons with this is it's not as cost efficient. It does cost more to do this type of extraction, and it does lend to some potential well-to-well variances depending on the analyst that may be doing the assay. Another pro that would be of this is it does work with a host of types of molecules all the way up to, siRNA. So when initiating your method development after going through your tuning and choosing your sample preparation, along that you should be studying stability. And that stability should be assessed in both the sample matrix and your stock or spiking solution diluent. So assessing both benchtop and freeze thaw is a must. It's a part of a validation test, but you should also be looking at exposure to light. There are times when you are unaware that light conditions may be affecting the sample themselves. So we do like to go through a light test of using both intense, visible, and ultraviolet tests in order to ensure that our compounds are stable under those conditions. If not, then obviously you take the conditions to ensure that they're not exposed. We also would look at oxidation. So this is kind of an unknown test, but it's something that does creep up when you have potential productions of oxidations with the amine groups, so N oxides and whatnot. We often see those off in the selectivity tests versus the general pools. So if you see where your selectivity is coming in with a negative value and your general pool is not, I would look towards putting an antioxidant into your matrix and seeing if that would solve the problem. Assessing stability in solutions in your diluent, as early as you can start that, the better off you are. So moving into validation, if you can have solution stability, that use of those spiking solutions will be stable throughout that validation rather than trying to establish it at the end. A big confusion is that when you see a drop in your solution concentrations, that it may be due to stability when it may in fact be due to absorption. So looking at well-to-well movement of your solvent to see if you get absorption is something that we do study on a case-by-case in order to ensure that if we have a drop, it's not due to stability, but maybe due to absorption. Another confusion is solubility. So a drop in your overall concentration, also being blamed on stability, may be solubility. So if you see stability at the lower end of your spiking solutions and the upper end seems to be dropping down, I would look towards solubility rather than stability in those cases. Finally, if matrix treatment is required, I would use the following guide. Make sure that the treatment volumes for the samples lend to accurate pipetting at the collection lab. So you don't want to be creating a treatment procedure where they're only adding 5 to 10 microliters of that treatment, because that can add variance to the samples. So working out that treatment is important to ensure that the pipetting is accurate. If you are treating preclinical samples, you should try and establish a treatment procedure that could also be used within clinical trials. So within the preclinical labs, they're very suitable to doing a host of types of treatments. But as you move into clinical, those full list of treatments aren't as viable. So looking forward into what could be used in clinical is extremely important. You should also be looking at matrix effects. So you should consider species matrix volume when deciding on preparation schemes. Things or species like mouse and rat have a limited volume, and they may require separate individuals for both high and low concentrations. As you may know, the ICH M10 has changed the way we look at matrix effects from the previous FDA guidance. So instead of 6 aliquots now, we're looking at 18 aliquots at 2 different levels. So those volumes can really impact the way you do that test. And you should try and prepare the matrix effects lots using the solutions used from your analytical curve. This is not a test of accuracy and precision, so you don't necessarily need to use your second stock solution used for QC preparations. Since this is a matrix effects test, you can use your analytical curve, and that will give you a more rugged test as to whether you're seeing an accuracy precision problem or if you're seeing a matrix effects problem. You should test hemolysis. You should do this with more than 1 lot of hemolyzed plasma within method development. That way, if both pass, you can basically state that you have looked at a rugged method. And it also eliminates if you only test one, you go in, it fails. You don't know if that's a selection of the lot or if that's a selection of the preparation process. The same things applies to hyperlipidemia. Really, you should be testing more than 1 lot. This is obviously for clinical assays. So a lot of the lots that we get within our house are extremely high within that lipidemic value. And you don't want the context of the matrix itself to really determine the outcome of the test. So testing more than 1 would give you that type of information. A couple things to consider: if your matrix effects fails due to precision, you may be looking at a matrix effects issue. And it's at this point where I would be looking at post-column infusion of the matrix blanks, where you're infusing your analyte after the column and injecting the blank to see if you have any suppression or enhancement along your source ionization. Because usually when matrix effects fails due to that reason, you see it in precision. If matrix effects fails due to accuracy, but you see precision across the lots, at that point, I would look towards preparation issues rather than matrix effects issues. So as you move from method development into method validation, we like to do a handoff to another scientist. There have been cases where a method development scientist has, produced a rugged method within their own hands, but as we move that into another scientist's hands, we start to see different issues. You really want to pull these issues out prior to validating the assay, because once the assay is validated, you basically landscape the yard, you built the fence. There's not much you can do other than going back, potentially changing the method, and having to go do other validation tests in order to run into sample analysis. You should really map out your analytical experiments prior to performing the validation, going through, figuring out which tests should be done where. You want to get the most critical tests done early, because if they don't meet the acceptance criteria, you don't want to continue on through those other tests. You want to deal with those failures at that point in time in order to ensure, if you have to go back to method development, you've created an efficient plan in order to do that. As stated, I would establish solution stability as soon as possible. You should also keep method development engaged while validating the assay. Really, the method development scientists should be looking at the validation work that's being conducted. Even if that work is passing, and it's not passing at a level that the method development scientist was seeing, we really want to address that issue then. If there has to be tweaks done to the assay at that point in time, you really want to address it early rather than after all the data has been collected and a review has been done. You should really QC in real time, getting folders from validation up through QC to ensure that things are being done properly is obviously important. And you should have an established guide to determine what the next steps are if a test fails to meet the acceptance criteria. You really don't want to look at this on a case by case. Well, it failed, so we're going to do a correction in this manner. You want to be consistent. You don't want to create separate rules for each validation that you're conducting. When you're moving from method validation into sample analysis, you want to conduct a method validation to sample analysis meeting to review the following. Look at the accuracy and precision data. Did you get good precision from your high QC down to your LLO QC? If there was a variance in that precision, what was driving that factor? Was it the internal standard? Was it the analyte response? You want to ensure that you don't take something that was hidden within validation into sample analysis. You want to update the method to include the established stability. So anybody that is looking at the method going into a sample analysis knows exactly how much benchtop or freesaw stability that they have. And if there's matrix stability issues, they understand the timeline that the sample analysis needs to be conducted. Update the method using the validation scientist as a guide. There may be wording that was put in place by the method development scientist that was kind of understood in a different way by the validation scientists. So looking at things from two different perspectives is always important to ensure that we get the most adequate description of what is being done in order to get the analyte that was within matrix into a viable extraction condition. You need to determine a reasonable batch size for sample analysis. We like to conduct as many samples in one run as possible. We often aim for 2 plates, but if we haven't seen the ruggedness, we don't want to take that into sample analysis and repeat sample analysis because we run into issues like divergent curves. If you are treating samples for stability, the sample concentrations for the study should reflect the untreated concentrations, not the concentrations in the actual treated samples. You need to keep the method development and method validation scientists engaged throughout the early sample analysis and conduct ISR as early as possible, even if there's only a small number of qualifying samples from run to run. I have never considered method development or validation complete until your ISR meets the acceptance criteria. So some common troubleshooting issues. So one, you cannot determine accuracy until you have precision. So if you're looking at a run and it looks horrible and you're looking to say everything fails, the first thing you want to look at, well, how is my precision? Because if your precision is not passing, you really do not know what your accuracy is. If you have poor precision, you really want to determine if it's extraction related or instrument related. So doing intra-vial injections of the same sample, if you have precision there, but yet you don't have precision in your QC sets, you've really determined now that it's failing due to extraction conditions. And at that point, you may want to extract some blanks, post-spike into those blanks and see once again if precision is there. If you don't have precision, then you're probably looking at a matrix effects issue. If you do, then you're probably looking at a recovery issue. If accuracy is failing, you should ensure that the preparation scheme for both the analytical curve and the QCs are similar. Is there same number of dilutions between the higher point of the analytical curves and the high QCs down to the lower part of the analytical curve and the lower QCs? You want to ensure that is not a stock solution issue. You can always prepare test QCs from your spiking curve. If the test QCs from your spiking curve meet the acceptance criteria, but the QCs from your QC spiking solutions do not, you're probably looking at a stock solution issue, in which case, go back, make all the stock solutions again, run them all for accuracy, determine where your bias is coming from. So if sensitivity is dropping during the course of the run, but returns back to normal when you put on a different column, you really need at this point consider adding in a forward or backward flush in order to re-equilibrate the column from injection to injection. If it consistently falls and a new column does not generate sensitivity, you may want to look at adding in a diversion or looking at different LC conditions so you get a more stable analyte response. So that conducts this early part of it. I'm going to hand it over to my colleague here, Corinne, to go through some spatial bioanalysis.
Thank you, Troy. We are now switching to the analysis of small molecule drugs in relationship to its spatial context and its added value. Can you go to the next slide, please? The plasma bioanalysis, it's an important aspect of drug discovery and development to understand the pharmacokinetics of a drug, its method of administration, and the dose necessary. But to understand the true efficacy of your compound, and therefore de-risk its development, tissue imaging analysis, referred as spatial bioanalysis, it's needed to answer, does your molecule drug reach its target? Does your molecular drug stay on target? And is the target engaged? All this answered at the site of action in preclinical setting to support administration and target exposure, interpretation of mechanism of action at the site of action for efficacy assessment, and supporting interpretation of safety result by spatial evaluation of drug and this metabolite. And in the clinical setting, to support the interpretation of efficacy by biopsy exposure evaluation together with plasma exposure. Next slide, please. Spatial bioanalysis consists of 3 pillars, including advanced image technology, in-context small molecule analysis, and use of artificial intelligence to increase the accuracy of its efficacy evaluation at the site of action. Next slide. One approach to spatially analyze your drug is the use of laser capture microdissection, where a selected region of interest based on histological or autofluorescence delimitation of the tissue is dissected, and quantification is further performed using LC-MS. Next slide. Another approach, which is going to be the main topic of my presentation, is to visualize the drug, is the molecular imaging with quantitative mass spectrometry imaging technology, or QMSE. Tissue sample from a whole body rat to core needle biopsy can be mounted on a slide and matrix sprayed. The sample is inserted into the mass spectrometer and the laser is shooting at every position of the tissue. One spectrum is recording for each position, which will eventually represent 1 pixel of the image. If you can go to the next slide. For all the individual spectrum, an average spectra is calculated and a molecular image is being displayed for every peak present in this average spectrum. The image on the right shows the high molecular diversity covered by mass spectrometry imaging. The main advantage are the specificity of this technology. Each detected neon one specific image. This is a non-labeled technique. It allowed the absolute quantification. Up to 1,500 molecules can be analyzed, and it has a spatial resolution that can go down to 20 microns. Next slide. Using this technology, we can evaluate the lifespan of the drug and its related metabolite by, for example, monitoring several organs at the same time. The example provided here is a typical early whole-body PK experiment in which the drug and its related metabolite can be spatially resolved on the image and therefore quantified separately in their spatial context to provide evidence of toxicology. PK is also calculated in different regions to support the target exposure in a clinical relevant region. Next slide. In collaboration with iTeos, we were evaluating efficacy of the drug epacadostat, which is an inhibitor of an enzyme called IDO1 for indoleamine 2,3 deoxygenase. IDO1 is involved in the immune system response and play a role in suppressing the immune system and allowing cancer cells to evade immune detection. The enzyme is involved in the conversion of tryptophan into kynurenine. Next slide. With the MSC technique, the heterogeneity of distribution of the drug in different treated tissue samples is visualized. Per comparison, epacadostat quantification on tissue using MSI was compared to LC-MS. Comparable quantity was detected with the two techniques. However, the distribution and accumulation in different histological areas was only perceived with mass spectrometry imaging. If you go to the next slide. The drug target, IDO1, monitored with immunostaining when overlaid to the molecular distribution of the compound, show an accumulation of the drug, specifically at the high concentration area of the target, emphasizing the fact that the drug reaches site of action for maximum efficacy. If you go to the next slide. Finally, it's interesting to note that the kynurenine-tryptophan ratio can be used as an efficacy tool in this instance. On treated samples, one can note a decrease of kynurenine and an increase of tryptophan at the emplacement of drug accumulation, emphasizing the ability at visualizing the target engagement for on-tissue PK at the site of action PK/PD assessment. The next slide, please. Here's an example of the study, this time with an Amgen compound that has been shown to have nephrotoxicity in the rat. And this particular nephrotoxicity was visualized first by histologic assessment. Go to the next slide, please. As visualized, lesion as degeneration and necrosis can be observed in the outer stripe of the outer medulla ossum in the treated rat. In order to study the impact of the drug in the appearance of the lesion, mass spectrometry imaging was used. If you go to the next slide. As observed, drug is widely distributed and not specifically localized in the region. In contrast, the metabolite M10, part of the Mercapture pathway, is specifically accumulating in the ossum area and possibly responsible for the lesion appearance at this histological region. This experiment showed the importance of measuring the toxicity on tissue and separate the drug toxicity from the metabolite. If you go to the next slide. Now we talk about the preclinical aspect to support the interpretation of efficacy in clinical setting. QMSC has also been adapted to work with clinical sample to evaluate drug exposure. In order to use, a clinical sample, the FDA guideline has been adapted to QMSC. If you go down, Troy, I guess, there's a little bit of a hitch on that slide. It doesn't show the whole slide. Yes, thank you. Specificity has been evaluated for analysis by QMSC. Stability as well has been evaluated for use in QMSC. So if you go to the next slide, we are performing as well sensitivity analysis, accuracy and precision analysis in order to show the use and the robustness of the use of QMSC in clinical sample. And I'm going to give you an example, which is the next slide. And here we are for tissue analysis and quantification. We're looking at skin punch biopsy. The concentration can be measured in the testis sample and as well per region of interest. So here we're looking at the skin punch biopsy when we have epidermis and dermis quantification of the drug. And you can see in the table below the different quantification of that compound for different patients. On the right are the QC, and there has been on the same tissue section, on the same slide for analysis at the same time than the tissue, the clinical tissue sample. And you can see that the QC are pretty robust in terms of their precision and accuracy. And I think I'm done. I think it's you, Troy.
Well, we're going to obviously take some questions, but before we get to those questions, we both would like to thank, our method development, method validation, our operations, all of our principal investigators, everyone who really supports all of the work that we do from the lab all the way up through final reporting.
Thank you both for that great presentation. A few of you in the audience have already submitted questions, so we're going to jump right in. And the first one is, what is the sensitivity for a typical QMSI experiment? Who wants to take that?
So I'm taking it. We're looking at a sensitivity that can go down actually to 30 nanogram per gram, but in average, we are between 100 nanogram per gram and 1 microgram per gram.
Thank you. And the second one is related. Can we use QMSI in large animals such as monkeys?
So not on the whole monkey, but we can definitely perform, a different organ, yes.
Great. Someone else has asked, what is the applicability of both techniques to proteins and biologics? I assume we talk about the different techniques that you discussed in your respective presentations.
You can take it, Troy, if you want.
Sorry, you ask that again, Andrew?
Yeah, sorry. What is the applicability of both techniques to proteins and biologics?
So LCMS, bioanalysis can be done with proteins, biologics. We're working more towards RNA, but it really works with smaller siRNA. For proteins, the typical path would be to look for a signature peptide. So we wouldn't be looking at the intact protein yet. Things have been established, to see intact proteins with regards to mass, but the sensitivity of those assays and the robustness of those assays really haven't met the criteria to get to full GOP analysis. But certainly a digestion of those proteins down to a signature peptide has been established, and we have conducted that work routinely here at Illyri. siRNA, all the way up to something that's maybe a 50 mer has been established with the LCMS approach. Getting beyond that, we are working on that. We haven't had really full-on validations conducted, but hopefully we can get to that point within the next couple of years.
Great. The next question is, what is the typical study design for a pharmacokinetic study in an animal model? Quite a broad question. Who wants to take that?
No, usually we are taking six animal treated and 1 control.
Mm-hmm. And how much more expensive, someone else asks, is QMSI against the traditional assays with LCMS or MS?
That's a difficult question. I don't know.
So, really, that's going to be dependent on the organ or tissue that they're going to want to analyze. Sometimes, the spatial bioanalysis may produce more detailed information. So even if there is encountered greater costs there, the data that you generate from that may lead to higher success of your drug development. Sometimes just going straight up LCMS homogenization approach may fit your purpose in terms of just getting overall drug concentrations from those tissues. But I think the intact analysis of those organs and that detailed information can be really crucial with regards to how far did your drug impact the target organs. So although you can't really get down to the dollar value of, what is the cost up front, what's going to be the savings on the back end? How far do you get into your research without knowing that type of detailed information?
Great. Simpler one now. What's the limit of the size of molecules that can be detected by QMSI?
We can go up to 3 kilodalton.
Mm-hmm. Another one is, I'm not sure if there's much detail in this one, but hopefully you'll understand and be able to answer. How many animals per time point is the question?
Typically, it's 6 animals per time point. And sometimes they'll be broken into both male and female. I'll state I am not a PK expert, so I'm more on the analysis side of it, but that's typically the designs that we see coming in.
Great. One that's just come in from the audience now. Can 3D analysis be used for just like QWBA, meaning quantitative whole body or autoradiography, to analyze for drug distribution in the whole body of a rat?
That's true, but it's a label-free, like technology in the QMSI, so it's not, like as expensive as using, like a radio label approach, and this is something that usually you keep, like for patient and human.
Right, and the last one we have, as it stands, there's 2 parts to this. First, how is dose selected from non-clinical studies for the first-in-human studies? And then how does PK/PD modeling play a role in dose selections or go in terms of go or no-go decisions? And what are the key features that one should consider before dose selection?
So I'll be honest with you, I'm not an expert on that question. I don't think I could give you the most accurate answer. Corinne?
Not really myself either, actually.
Okay. Well, on that
but we'll come back actually, like with the answer.
Sure.
Yeah,
so
we do have people that can address that question. It's just not us two that are talking today. So we can certainly take that in if you want to submit it. We'll get back to you.
Absolutely. Yeah.
Yes, obviously, yeah. The person who posed that question, they'll get back to you. So on that note, we've run out of questions and we've also run out of time. So I'd like to thank our speakers today, Troy Volker and Corinne Ramis, for that great presentation, and also thank our sponsors, Alere Bioanalysis, for making this event possible. On behalf of Alere and Sightline, have a productive remainder of the day, and thank you for watching. Goodbye.
Thank you.
Thank you. Bye.